I will start now. So there are around, I think, eight of seven of you. I think, yeah, hopefully people will join as we go on. I hope you had time to go through some of those videos for the week two, right? So in that case, I will go through some of those concepts. I can help you understand those concepts better, and then we'll try to solve of some of the problems associated with those concepts. Okay. Please do try to interact because I would like to have this as a much more interactive session rather than me teaching you just listening. Is joining this session necessary? No one is asked. No one. You're not forced to join the session, but if you want to learn something, you are welcome to join the session. So that's how these things work. So yeah, it's upon to you if you whether you want to learn or whether you have other important works to do. Because I'm also giving a lot of my work trying to make this presentation and then spending 2 hours with you to help you understand these concepts. So hope that answers it. Okay, let's start. So hoping that you guys have watched the week two lectures. Can someone say what does Simon's hierarchical principle talk about? If you're not willing to speak up, you can write in the chat box also, which is completely fine. I can read from there. But yeah, it will be nice to have it as an interactive session. Hierarchical structures are stable. Yes, hierarchical structures are stable. But can you explain what are hierarchical structures and what is Simon here trying to say? So, if you have watched the video, so it's there. This concept of hierarchical principle is explained with the help of the two watchmakers example, where the structures that have this hierarchical nature tends to have much more stability the across the count across the levels. Like one of the watchmakers who has to assemble all of the pieces together all at once versus the other watchmaker who has smaller assemblages set up together, which together form the bigger piece, and finally the watch has much more advantage over the person who makes it all of it together all at once. So in that aspects, hierarchical structures are stable. So this principle mostly talks about, in natural world, and you have examples in artificial worlds also, speaking mostly about how complex systems are often hierarchically organized. Like they are like, subset of subset within each other, or smaller puzzles put together form the bigger one. In other words, they tend to be structured in layers or there are multiple levels. So this is captured by this principle, and then these structures, what's the advantage having such so many levels or layers? So because the major advantage is these structures are much more stable across time or than those which are not layered. And using these stable structures, we have something called emergent principles, which which suggests that the sum of parts or the whole, when you just look at the individual parts of these structures or of individuals, they tend to have, they provide different things. Let's say, for example, let's take an ant, which can bite when it's going out to look out for food. it can, let's say, it can get 1 grain each. But if you the whole colony, as you know, there, let's say there are 100 ants in the whole colony. So the 100 individuals can transfer, much more faster or efficiently than the compared to individual ants getting them together. So these are some of the ways these emergent principles are there in nature. So basically, yeah, some of the parts, individual, the whole to kept together, it performs better than the individual parts. And the whole parts put together has some other unique properties that the individuals themselves don't have. Okay, since we are talking just about ants, the picture that you're seeing here is the picture of an army ant, mostly found in Amazon rainforest. Okay, so the beautiful thing about these ants, so these are individuals as you're seeing. So, but the individuals, when they come together, they form these beautiful structures which they call themselves, they use as homes because they don't have a permanent home like other insects or other ants. They make homes out of their live bodies itself. They just simply hang hang on to each other legs, forming these homes. And they just simply the whole colony and have a resting place for the queen and the workers on and the larvae. So yeah, and then some of the functions the colony together can do can't be done by individual workers. And biological organizations. So this is something which we all are familiar with. So it all starts with a cell, a group of cells form into tissue, and then such tissues, similar types of tissues are forms an organ, and multiple different organs forms organ systems, and organ systems kept together forms, an individual or a species of whatever you are looking at here. It can be kangaroo or rat or plant, anything. And then this is at the individual level. But when you have such individuals, it forms a population. And then different populations of multiple species. As you can see, there is a deer, hare, there's also multiple species of different populations form together a community. And different such communities are in the are together form are part of an ecosystem. And different ecosystems are part of a biome. And then different biomes are a part of constitute the biosphere. This is what we see. And if you remember from the previous class, we talk about different type of ecologies we have. If you're looking at individual species, you call, species ecology, population ecology, community ecology, and so on. So if you look here, if you look at the ecosystems which we are seeing, can someone say what all these ecosystems are? Like one, two, three, from the left, starting from the left. Can someone say which ecosystems are these images roughly representing? You can write down the answers in the chat box also. So what's the one on the left representing first? Grassland ecosystem. Yes, nice. That's a very specific answer, but yeah, that's the right answer. But I was looking out for it's part of a terrestrial ecosystems, and the one in the center is is an example of freshwater ecosystem. It's a riverine ecosystem. You have freshwater, you have pond ecosystems, lake ecosystems, and even riverine ecosystems comes under first freshwater ecosystems. What about the last one? What do you think is the last one representative of? Coral reef, yes, these are coral reefs. So that image was supposed to represent marine ecosystems. So these are the major group of ecosystems that we see, and then we have subsystems like yes, aquatic. We have two different types: freshwater and marine. So under these marine ecosystems, we have different type, or even terrestrial. We have grasslands, forest ecosystems, or mangrove ecosystems, and so on. Okay. So, and then similarly, if you go back to the slide, we have multiple ecosystems kept together forms a biome. So, how are these biomes classified? So, on what basis? So, if you look at this plot, most of the biomes are classified based on the annual temperature and then the annual precipitation. So, annual means the precipitation that happens across the year. So, let's say, for example, let's say tropical rainforest, which everyone might be familiar with, where there is, annual precipitation is much more higher, and then the temperature annual temperatures are all on the higher side. But if you go to deserts, temperatures are higher, but annual precipitation is much more lower. But there are cold deserts also, where the temperature and the precipitation both are very, very less. And then you have temperate rainforests, tropical season, savannas, woodlands, various different types of biomes that come in between. So mostly it's the precipitation regime, basically the amount of rainfall and the temperature that governs the distribution or the type of biomes that you see in an area. And then if you look at this plot or the map here, so these are the major biomes that are present across the world. These are further classified than what have seen in the previous slide. So the ones that are highlighted in the red are the ones that can be seen across the India, mostly the temperate broadleaf forests, which are somewhere around, you know, here in the Himalayas. And arid desert you can see in Rajasthan, and tree savanna, subtropical dry forests, and tropical forests, mostly in the pen- tropical forests in the Western Ghats and the northeastern part of India, and subtropical and dry forest here, and savannas here. And then alpine tundra and mountain forests are also in the Himalayas. These are confined to the high elevation areas in the Himalayas. So, and there are species that are unique to these different biomes. Okay, so what do you think of this word biodiversity? When you hear this word biodiversity, what do you think of it represents, or what do you think it says? Variety of life. Yes, yeah, that's true. Any other people want to add anything? Variety of life. It's it's a shorter way of saying what is biodiversity, easier way. Anyone wants to add anything to it? Okay, I'll take it as a no. And so the term biodiversity also refers to biological diversity, saying that different variety of life on Earth, and this diversity can be at all levels. It can be at the level of genes, which are which are the smallest unit you can think of as of now, to ecosystems, which are the the largest levels that we have seen earlier. And this can also encompass different processes that sustain this life, that this variety of life that we get to see. So we have so this diversity can be either species diversity, as you can see different species of ants present. So it can be birds or plants or anything. If you're just looking at them at the species, you call it as species diversity. Okay. What do you call this as the variety of living things and the interactions? Yes, that was right. So that was biodiversity. Yeah. Now, can someone say what do you think you are seeing in this picture representing? GM plant, might be GM. I'm not sure about that, but what do you see? So are these of same species or not? Then let's go slowly. Are these same species or not? Like, are they different things or are of the same thing? Yes. Species. Oh, it's not species diversity. Yeah, everyone is agreeing that it's the same species. So, but what you're seeing, there is a diversity in colors, right? Some are yellow, some are black, red. Some are there's a mix of bunch of different colors. Genetic diversity. Yes. Yeah. Someone said Indian corn, American seed. Oh, that thing I'm not sure. I don't think it's that. But there is it shows it's all of it is the same species, but you are seeing diversity at least here represented in terms of colors, which talks about genetic diversity, the diversity this different individual varieties have in their genetic makeup. Okay, so there are multiple levels of diversity, but I was just going through what is species diversity. Basically, you are looking at individual species together, and genetic diversity is within a species. How diverse are the genetic makeup of the species? Okay, for example, here we took the example of corn and sales, different varieties together. And can someone answer or think why should we measure biodiversity? Any anyone? Why do you think we should measure biodiversity? Like, why can't why not just say "Okay, it's there? So why should we bother about it? Why should go and take the hassle of measuring, going to these remote places? No one wants to answer this. So there is no single answer to it because everyone has their own perspectives. For example, if you think in terms of a conservation person who wants to conserve a species, so or an area, so they wants to measure the biodiversity of that area so that they can have better understanding of what all species are present, what are are rare species or that are important species that are need to be conserved. But if you talk with respect to high biodiversity is perceived as a synonymous with ecosystem health. Yes, that's a really interesting point. So in some areas where there is high biodiversity, you say that the system is healthy, and then it's perfectly good. And unlike there's some of, like, for example, if you look at a forest versus a site that is close to a city or a park, you know the biodiversity of these two places are entirely different. In those aspects, you can say the high biodiversity that you're seeing in forest, they represent that area is healthy, not as much as a city. So that's a really important thing. And then along with that, some people may have wants to understand what species of medicinal plants are there. Okay, so so that humans can use those some of those medicinal plants to cure some diseases which we are not able to find cure as of now. So some of those things for you to identify, you need to have an understanding of different aspects of biodiversity. Okay, so to do that thing, we need to have one of the basic things. The first thing that we go through is like documenting the species, and then once you start documenting the species, you try to understand this concept called species richness, basically which means the number of species present in an area. Okay, so here, as you can see, some like a researcher has gone to identify the plants in an area, let's say two different forests. One is, let's say, one is in Karnataka, the other one is in Kerala. And then he put up some plots, like 10 by 10 centimeter plot, 10 by 10 meter plots, and wanted to identify what all species are there. One, what is the species richness of the two different plots, and then he wants to see which site had higher species richness. Now, for example, if you can all of you guys go through this example and say what is the species richness of the community one and community two, or say which community has higher species. I can tell you how to do the evenness little later, but species basically, can someone count how many species are present in community one and community two and say which community has higher species? Two. Community two, or the answer is two. The number of species, the shape, color, you can look all those things and then say that is a different species. Okay. For example, I can help you with one. Let's say this is one species. This is another species because there's shape. The shape is different, and the color is different. I think one. The answer was two, which has the highest. Okay, I think you're answering for which community has highest species. Okay. Any more answers? Can someone or someone wants to take little time to solve it again, or you can take a pen and paper and try to solve it yourself slowly. Community one. Okay. Mrinal said community one is even. Okay, we can come to evenness later, but we are currently at species richness. Which community has higher species richness? Community one, community one, community one. Okay, so for people who are not able to understand, let me help you solve this. Okay, uh, so different type of species. Let's call this, some looks like a conical shapes. We'll for now call it pine. Okay, so there is one pine species. There is some other tree species. This is two species too. This is different color, different shape than this one. Okay, everyone agree this is different species, right? And then this is another species which, shape and color are different. And then the one in the corner, this is also entirely different. So as you can see, can you see any more species? Like, here I can see one, two, three, four, not more than four different types. There are four of this dark green, four of this light green, there are four of this, and then four of this. So the species richness is basically the total number of species present in that community. So in community one, it is four. Okay. Uh, just give me a second. Yes, it is four. Okay. But if you come to community two, let's do that thing again. So we have this one, this is two, this is three, okay, and then this is four. Okay. So the total species that you can see in this community is also four. So are the communities, so both communities are, equal in terms of the species richness. One of the metric that we said key to differentiate different species, different communities here. So both are, same. It's not like different, because the total number of species are also four in this case, and also four in this case. But okay, so do you think these community one has more type of trees? Someone said. Everything is a tree here. Yeah, we should not get confused. Everything is a tree here. So both have the total number of, species is four, which is like both. It says ki both communities have same number of species. But do you think both communities are same, the community one and community two, or are there any other differences in those communities? Differ in evenness. Yes, you're absolutely right. So, can Prashanta, can you just write or say what do you mean by evenness here? So, what are they differing in? So, yeah, even if you're wrong, I can help you correct it. So, don't worry about it.
Yes, in the first community,
Yes,
there are four of the species are almost equally distributed. But in the
yes,
second community, one particular tree that is that I think the shape is you have written there are two. The
yes,
number two plant is more dominant. So that is the
yes,
dominant species. So
yes,
that's why the diversity in community two is less as compared to community one.
yes. So yeah, you're right. So in community one, every species, is more or less present in equal numbers. Let's say, for example, let's count the number of dark, these conical shaped trees. One, two, three, and four. That these ones. One, two, three, four. So each species is present in equal numbers here. But if you come to community two, as uh, Prashanta and Mrinal has pointed out that one species, the species two, which you are saying, is present in huge numbers like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 11 individuals, which is like is the only most major species or dominant species present in the community, while other species are present in either one or two numbers each. So these communities are not the same. So they are different. So what they are different in? They're different in their evenness. So if you calculate, so the proportion of the species is 25, 25, 25, 25 here, while here this species has the greatest amount of proportion because it's dominant and it's present in huge numbers in that area. So we call this community is not as even as this. Okay. Compared to this community one, community two is not even. Or compared to community two, community one is more or less even. Okay. So we understood how species richness and then is measured, and then how the evenness is important to understand, some communities. Even the species richness is similar, the communities might be different in terms of the number of individuals of a species are present in that area. So that's why understanding species evenness is also important when you try to understand the biodiversity of an area. And so to incorporate all of this together, so there are different diversity indices that has been proposed, to that incorporates both richness and evenness, because richness, and as we just learned, richness alone might not be able to capture whatever the thing that is happening in the area that you are studying. So you need to have consider both richness and evenness. So the two indices that capture these, one is the Shannon Simpson's index, the other one is Shannon Weiner index. Okay. There are multiple the denotations for each of these index. For Simpson, you can have D, lambda, or HS. While Shannon Wiener index is also called Shannon's index. It's H or H hyphen slash. I don't know what. Yeah. So the main difference between these two is uh, in Simpson's index, both richness and evenness are considered, but compared to Shannon, the evenness has much more influence than the richness aspect. So when you try to calculate it, if the community is more even, it might have higher values. But in Shannon Wiener index, there is no higher priority given to either richness or evenness. Both are considered more or less similarly. And once you calculate the Simpsons index, you just subtract it from one, to get the true value of diversity. So the formulas are these. So don't get scared by these formulas. I can just explain. So if you just look at Simpson's index, it is saying sum of pi square. I is equal to 1 to r. Basically, you sum all the species I till how many species you have. Pi is proportion of individuals. So let's say, for example, if you go back to this example here, what is the proportion of individuals? How did you calculate here? So let's say we call this species. So this species has, four individuals. Out of total 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Yes, 4 out of 16. Okay. So that is how you calculated the proportion to get that 25%. So if you do the same thing here, we have, this species has two individuals. 2 divided by 16, right? So that's how we calculate the proportion. So that is what this PI says. So like the proportion of the number of individuals of that species divided by the whole number of species, total number of species present in that community that you have sampled. So that is what divided. So n, the small n is the number of species that you are looking at currently, and the total number of individuals found of all the species. Similarly, here also the pi means the same. Okay, but the thing that you have that come across the other thing that comes in this Shannon's index is we have log, natural log ln that comes into Shannon index. Okay, uh, r is the same thing. Okay, just a minute. Someone asked a question. Simpson's index and SAS. Simpson's biodiversity index. Yeah, they are. You just add multiple names to it, but both are same. So yeah, there's no much difference. But the way they're represented might be different. So don't get confused. Sometimes instead of lambda is equal to sum of pi square, they will just show one minus sum of pi square. Okay, so just don't get confused. They both are the same. Okay. Uh, that other reason, the other thing is in Shannon Wiener index, you subtract the whole value with a minus. You'll soon get to learn why we do that because we get the values in sometimes in minus. So yeah, to get a positive value, you just subtract. You use minus. Sorry. Yeah. So now can you guys use these formulas? Sorry, minus is for multiplication there in Shannon's index. Can you guys use this formula and calculate, Shannon's diversity or Simpson's diversity index, whichever you can, whichever you want to, so that I if you guys get it wrong, I can help you, do the same thing again or tell you how should it be done better if you are not able to do. Why Simpson's index is 1 minus d and Simpson's biodiversity index is expressed as b. Uh, so I'm really not sure, but in the end, when you tend to report, so it's basically just when you look at Simpson's biodiversity index, it's the value. So you get some values, right? So the values are in Simpson's diversity index mostly constrained between 0 and 1. So the lower the value, uh, it's greater the diversity. So and the higher the value is, less diverse that area is. So to prevent, I think to prevent uh, that confusion, you tend to subtract one the d value -1 so that higher the value of 1 minus d, you have higher diversity. The lesser the value of one measure, you have low diversity. I know if it might have just gone over your head, but if you can calculate this Simpson's index, I can in the subsequent slides explain why 1 minus d is used to calculate the final index. Uh, are you guys willing to solve this thing? Can you just let me know this? Or if you're started solving, you can go ahead. I can explain for both of these indexes how can you solve. If you want me to show, I can show that slowly, but I would like you to give it a try in solving. Okay, tell us the number of different species. Okay, I think Prashant, yeah, I need your help to do this. I can, but yeah, I don't want to do it myself, but I want you to learn how to do it. So you help me. Okay, let's count. Can you unmute yourself and then join me in identification?
Yes.
Yes. So let's count the ones with the long tail. Okay.
Okay.
In Lake A.
In in Lake A, there is only one pintail.
Okay. Are there any more? Lake A.
No, in Lake A, only one pintail.
Yes. You just name it. Long tail. Just name it long tail or long tail duck or something. Just write it
Okay.
down as long tail duck. How many individuals? One. Let me
One.
just write. I'm just writing it as LT. Okay.
Okay.
LT. Number of individuals. One. Okay.
One.
Now let's go for the easier one. So the big. Let's go for the bigger one. Okay. This is species two. Okay. The
Species two.
yeah, I'm writing as BG, which is so big. How
Okay.
many individuals are there?
Only one.
One, right? Other
Okay.
people can also join if we do mistakes. So please, and let's go for the one with white ear. Okay, let's call this species three. So like I'm just writing WE. Okay, how many individuals are there of white ear here?
I think only one.
Only one, right? Yes. Now we'll go for white belly. Okay, WB
Okay.
because it has a whiter belly. Okay,
Okay.
how many
It's
individuals with white belly?
it is also one.
One, right? And the remaining ones are there any more species or is just the same? The remaining ones, the species five.
Six, seven,
There is no other right other than five. We have just five species in this, right?
16, 17.
17 individuals. I'm just naming it
17.
black duck. Okay, BD. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20.
1, 2,
Am I right? The black duck, this one is 20 individuals, right?
3. I have counted 17.
17. Can you do it again? Because I think I counted 20.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Yeah, it's 20.
Yes. Chalo. Okay. Now we have the number of individuals of each species of this. So if you do the total, what is the total? Total T, how many individuals? 20, 21, 22, 23, 24. Okay. Okay. Similarly, does anyone wants to do it for leg B? Help me with leg B. So LT, we have the same things. LT, let me just change the color.
1, 2, 3, 4, 5. 5 birds.
Five birds. Okay. What about BG?
It is also same 5.
Five. Okay. Next. WE.
White ear.
White, white ear. White ear, I don't see anything. Do you see anything?
No, nil.
No.
Zero.
So zero. Or we can just, leave it as blank. We don't
Yes.
need to write that at all. But I'm just writing it. So what
Zero.
about white belly? Do
One,
we do you see any white bellies?
three, four, five. Five.
Three, four, five. Yes, five. What about big duck, black duck? The small these ones.
It is also five.
Five. D is five. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So the total is 20. Yes, right. So now the total is 20. Okay. So I'll just write it again here. How to calculate PI? Okay. PI is proportion. Okay. PI equal to, let's say if you want to calculate the proportion of uh, LT, which is long tail ducks in lake A. So how do we calculate? 1 divided by total number of individuals in the lake A. So that is 24. So whatever that value is, you will get. Let me just do that quickly. 1 divided by 24. It's 0.04. Okay. So that's similarly, you can have another column in the in the notebook you are doing. Write it PI individual of each of the species, and then write it. And then the other column you can square it, and then add it. So whatever the value you get should be the Simpson index. Uh, is this fine? Can you do it from now, or you need further any more help? Okay, sure. Take your time. If you are stuck anywhere, just let me know so that I can help you through. Uh, Answer is 0.99 for just write the which lake you are saying that answer is. So you have to calculate whichever diversity index you are calculating for both Lake A and Lake B, so that you can compare between those two lakes. If you just have for one lake, you can say "Okay, this is the diversity, that's the end." But if you are trying to compare it, which is what we are trying to do here, we need to calculate the same thing for both the lakes. So then you can say based on this value, this lake has higher diversity or this lake has lower diversity. So when you're writing in the chat, just write lake A, what's the value, and then mention which index you have calculated. And similarly for lake B. So if you have a book, it gets easy because you might forget some of the numbers when you're doing. So you can remember using your mobile calculator if you have adjacent to you. 0.69. Okay. I think I will also do simultaneously with you people so that we can check our answers together. Lake A 0.69. Okay. Shannon for Lake A 0.7. Okay. Okay. Do it for Lake B also so that we know you can compare, and then once at least few people have done it, I can go about explaining the answer slowly. I think that is okay. Uh, okay. Prashanta has calculated. So is anyone trying to do Shannon's diversity? Okay, sorry. Simpson, has anyone? Mrinal, can you just write in which measure you used? Okay, Simpsons. Has anyone other people have done? Please write your answers. We can wait for five. I will wait for 5 more minutes so that you have enough time to do. So other people who have already calculated Shannon, if you want to try out calculate the other index, you can also try and calculate it again. Prashanta, you have calculated Shannon, right? Simpson for Lake A 0.68. Okay. So any other people have tried to calculate it, please try to write your answers in the chat box so that if there is anything that you're not able to understand about these calculations, I can help you slowly so that you can do it better. Okay, so should I move on to the next slide and show how for each of these indices for these two different lakes? Okay, should I or should I wait for you to calculate in want to compare your answers to the ones that I show? Okay, I will take it as a no and then I will move. So if you see here, as we have said, yeah, species one, 2, 3, 4, 5, 1, 1, 1, 1, it's 21. I think it's 20 only. So yeah. So once you have the counts for the species, you know, uh, what we do is like calculated the PIs. So this is basically the PI. So the proportion of individuals compared to the whole total number of individuals in the in that community, and then we got 1 out of 25 is 0.04. Let's go back and just count it again. Where 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 19, 20. No, yeah, I think the example might not be in right one, but there are just 24 individuals. So it's not 21, it's 20 here. So what happens is 0.04, 1 by 24. So a lot of you might have also got somewhere around 0.04. And now you do this PI for, all of these, all the species till here, till the 5 species. And then you square the values, PI square. So 0.04 square is 0.0016, right? So once you have these PI squares, you just have to add it up. So when you add all of these, here you are getting 0.71, but you will get somewhere around 0.69, so on, or zero point 0.7, which I hope some of you have got. I think, Prashanta, you said zero point 0.7 for Shannon. Lake A, is it Shannon or Simpson? I'm not sure. Okay, Simpson 0.68, you were saying. Okay, that's close. And even Mrinal said uh, 0.69, which is you're close. So, which is also the actual value. Here, don't, here is, if you do it with 24, you will also get some same value. Okay, and similarly for Lake B. So, Lake B, when you do this for proportion, so basically we had 4 species. We didn't have one of those species, so we can just, this is not there, but if you just calculate 0.04, 0.04, 0.04 into 4 times, what is it? Let me just a minute. 0.04 into 4, it is 0.16. Okay, 0.16. Okay, so the whole D we got is 0.16. Here, what we got here is 0.77. Okay, so this is D values. But if you want to calculate, since I was saying earlier, the diversity, you know, as you can see, there are 21 individuals of this species, but you are getting, 0.7 value, which is like higher value. You know, this is, a case of low diversity. But, here high diversity and high evenness, but your value is so small. Basically, for you to interpret it better, what we can do is like we can subtract what the value of D you got from 1, so that you get, what you get is final value here is 0.3. Okay, and then what you get here is 0.84, right? So from this 1 minus D values, does it say? Lake B has, higher species richness, or sorry, higher species diversity than compared to Lake A, right? I think Mrinal asked this question. I hope this thing answered you. So why do we, matlab, why we do it 1 minus D? So it helps us understand it better instead of having confusion, right? So this is Lake B has higher diversity. If you go and check it out, the previous example also, if you remember, we said it's not only the species richness, the number of species present in that area matters, because the Shannon and Simpson take take in consideration both species richness and evenness. You get those communities with higher species richness and evenness as the ones that are much more diverse. Even the species richness is higher here, we say it's not even. So we say this community is not diverse. Okay. Do you have any doubts still here for this Simpson index calculation? Because yeah, if you have, do let me know so that you get clarified. It gets clarified here itself. Uh, Prashanta, where do you think have you done wrong for Lake B? You got one minus 1.38. Lake 2 is 20 individuals. Oh, sorry. Thanks for pointing it out. So it will be it's 5 by 20, right? 5 by 20 is 1 4 is equal to 0.25. Okay. 0.25 into 0.25 is 0.625. 0.06. Just a minute. 0.25 into 0.25. 0.0625. So, okay. It's my bad. I forgot about that thing at all. It's 4 are there. It's 0.25. So the final value what we get is 0.25 here. Okay. Kindly ignore 0.16 here. So it's 0.25 what we get. So once we subtract this from 1, what you get is 0.75. Okay. Uh, so Prashanta, I think just have a look at this example because this is how we calculated it. Just you can go around when later on once you get free time, have a look at where you might have done wrong, because it is you just had to have these proportions and then multiply it to get, and then add it up. So 0.625 into 4 times is 0.25, and then once you have that value you can just subtract it from 1 so that you have the final diversity index. So now Lake B has higher diversity compared to Lake A, which has lower diversity. But the species richness is reverse. We have 5 species in Lake A and just 4 in Lake B. Right? I hope this is clear. Does anyone have any more clarifications with this diversity index? Since very few of you are here, feel absolutely free to interact or ask anytime, because I can get back to each one of you again and again if you don't understand. Okay, okay, nice. Glad you understood it. So I will go for Shannon diversity next. So similarly, in Shannon, what do we do? So let's go with the example here. Let's consider there are 21 individuals here, so that the calculations of log pi gets easier. But if you want, I can calculate it. Just let me know. If you are not able to understand, we can go back to the same example and do it. So uh, it's 20. We're just going with 21 as of now. So what do we do here is like calculate the proportions, like what we calculated earlier is like 1 by 25. In this example here, 1 by 25 is 0.04. Then you take a natural logarithm of this value, okay, log of pi. So which gives log of 0.04 gives minus 3.214. value. So minus 3.219 into 0.04, you will get 0.129. Similarly, once you are done it across for all the species, you just simply add it up like the previous case. So you get, you know, some value like this somewhere. So the value says 0.661. Similarly, for lake B, we also have a value of 1.61. Okay. Uh, okay. 0.6. I think Prashanta, you did. You reported in the chat. I think reverse. I think you said for Lake it's 0.68 Simpson, but I think you might have calculated Shannon's for that because the values are matching here for 20 individuals case, 24 individuals case. So yeah, so what do you think? Which now we have these values, right? 0.66 Lake A and Lake B has 1.61, the value of Shannon index. Which lake do you think is diverse here in terms of the birds that are present here? Lake A or Lake B? Lake B. What do others think? So one person think it's Lake B is much more diverse. What are other people? What the is Lake B or Lake A? Uh, so yeah, you are absolutely right, Prashanta. That is Lake B is higher diversity. So the higher the values when you calculate this index, you can just say that the greater is the diversity of the lake. Okay, here the birds in the lake. So yeah, so but for Simpson to say that the higher the values, the higher the diversity, you have to do this calculation of 1 minus d. Okay, can you see any other relation between Simpsons and Shannon, at least in the diversity values that we are getting? Uh, so for example, if you look at Simpsons index, it will be much more constrained. You get values either from 0 to 1. That's it. It will not go beyond 1 or below 0. But if you look at Shannon's, the values often exceed 1. Okay, so just keep that in mind. So this is, once you add all these things, you have to multiply the whole value with minus, because once you take logs, your whole values are in minus. So once you add it up, this is for multiplication. Okay, so don't forget to multiply so that you don't have values in no minus. I think some of you might have reported the values in minus. Okay. I hope this calculation part is little clear, but you may not be asked to do in the assignments. I'm not sure, but generally this is how the calculations are done. If you end up with equation and said that the picture like we have seen before, you should be able to do this calculation. Okay. This is clear till here, right? Okay. In that case, I will go to the next slide. Okay. Yes. So, now we have seen how to, you have a little idea what is species richness, what is, how do you calculate uh, different diversity indices and say which community is much more diverse. So just keep that in mind that species richness is a different thing, species diversity is a different thing. Okay. What we calculated now was You can call it as species diversity. Species richness is the number of species. That's it. The species diversity is like how diverse is the community in terms of both the richness, evenness put together. Okay. Sometimes when you go out, uh, calculating the number of species, you get to you make this curve called species accumulation curve. So basically, this is a natural phenomena that you get to see, whenever whichever species that you're trying to look at. Let's say, for example, you have gone, if you have gone to an area, you know, and looking at you have gone to watch birds in an area. So if you look at, number of individuals on x-axis, plot the number of individuals you have seen and the number of species they belong to. So first 10 individuals you saw, let's say, are of, myna. So you'll just have one species for the 10 individuals. And the next 10 individuals you saw were of, parakeet and then kite. So the number of species also tend to increase with the number of individuals, as you see initially. And then after a while, you know, you have seen most of the species that are present in the area that you are looking at. So it tends to, stagnate. You hardly get to see any other new species that you are not seen at that time. So it kind of, accumulates and, and kind of forms, straight line. So those type of, so if you make a curve make a smooth curve, you call it a species accumulation curve. So basically it shows how the species are accumulated as you go on sampling for or looking out for more number of individuals or more time. Okay. So this is how the original curve looks like. But when if you try to repeat the same thing multiple times, you get, a curve, something that looks like this with mean value and then uh, mean and the associated standard deviation with it. So this mean curve is called rarefaction curve, or you can call species accumulation curve. This is the original curve, individual curve is species accumulation curve. You can call the mean values as the rarefaction curve. You can do it for number of individuals versus you can do it also for the time spent on you know observing birds in this example. Okay. These are especially important, in understanding. So this species accumulation curve plays a major role when you're trying to understand whether have you sampled all the species in that area. Let's say, for example, you went early in the morning to watch birds. You got 20 individuals here, and then you got number of species. If you plot, you're still here. You miss, another 15 species that you could have gone. So you had to, increase the number of samples to get to capture that. So saying that your sampling was the effort you have put in and finding out the species was good enough to capture the number of species present in that area. So sometimes you don't know the whole species, so you tend to use this theoretical approach to say that whatever the way I had collected the my observations were enough to say I have captured the most number of the species present in that area. So in those aspects, these species accumulation curves are important when you're trying to document biodiversity. Okay, uh, it's you could understand till here, right? So now I will go into the diversity matrix. Okay, so there are different diversity matrices are used. So some of the very basic matrices that we use is what we will do today. So the first one is alpha diversity. Okay, alpha. What does that mean? Alpha diversity means the mean species richness of a site. Okay. Uh, so if you say this as a site and this as another site, if you want to calculate, if you want to find out the number of species present in each site, the value is called alpha diversity. So what is the alpha diversity of site 1 and site 2? Can you please write down the answers in the chat box? What do you think is the alpha diversity of, you know, site 1 versus site 2? 4. Okay. What about the other site? 4. 4, right.
Four,
So,
sir.
yeah, both the sites have, the total the species richness as 4 each. Okay. But what do you so okay, let me just write it up. What you just said. So it's 4 each, right? I'm just writing the average for both. So gamma diversity is the overall diversity of all the sites that you have, gathered the data for. So let's say you went to a forest, you put up two plots and then sampled, trees present in them. So you found four trees each in plot one. In plot two, you found, another four trees. But if you're talking about the total number of trees that you came across in the forest, you had to, include all of the species that are present in all the plots that you have sampled. Okay. So what will be the value of it then? The gamma diversity. Basically, of all the plots of the area that you are looking at. So if you want to know the gamma diversity, let's take it to a higher level. 8. Are you sure is it 8 or are you asking me whether it's 8 or not? So the answer is you're right. The answer is 8. So yes, yeah. Sorry for the messy way the 8 is written. The answer is 8 because there are four species here, and then there are, you know, four species here. So uh, which will, make the total species as 8 present in the whole forest plot, which we are discussing about. And there is an interesting metric. So this is just the species, the total number of species present in each plot. So the third thing is called beta diversity. Okay. So what does this talk about? This talk about how the species change between two different sites. Here, site 1 or site A and site B. Okay. How the species diversity changes or a species composition changes from community 1 to community 2 in the area. So there are multiple ways to calculate beta diversity, but I will tell you the easiest way to calculate beta diversity, which is you can divide the gamma diversity by alpha diversity. So gamma by alpha. So what will be the beta diversity of this area when you calculate it by gamma by alpha? Gamma by alpha. Can someone quickly calculate and let me know what is the beta diversity when you calculate it by as gamma by alpha? Okay. 4. How come it's 4? I'm just trying to understand. So what is the gamma value? So the gamma we have already done, which we said it's 2. Yes, Bala, you're right. Well, how is it 2? I will just explain. So what's the gamma value we have? We have 8. Yes. Uh, and then alpha is 4. So when you take alpha, you take the mean alpha, average basically. So average across the two sides, 4 plus 4 by 2 is the same. So average is 4. So the value is 2. The beta diversity of these two sites is 2. Yes. Yeah. Okay. So there are different matrices. So beta diversity here talks about how different, are the these two sides in terms of their composition. You get some value like 2. But this should not be confused with the species richness values, because this is an entirely different value. So it's different from alpha and gamma, but it uses alpha and gamma to come to beta diversity. Okay, so alpha, I'll just go over it again. Alpha is the number of species that you see in the smallest sampling unit, if it's a plot or a transect or a point or anything. However, gamma is at the higher level. So if you have multiple such plots kept together, so what is the total number of species that you are seeing all of those plots kept together? So that value is gamma value. And beta diversity talks about how the change of species occurs between the different sites you have sampled. Okay, so the way we have calculated beta diversity here is gamma diversity by alpha, mean alpha, or average of the alpha diversity across all the sites. Okay, so there are different ways to calculate beta diversity, but this is the easiest way. And then yeah, at this stage, this is what should do the job. And can someone speak about this slide? What are biodiversity hotspot or the areas that are shaded in red? Can someone please just speak voluntarily or write down in the chat? High species richness. Okay, that's one aspect. High species richness. Is it the only criteria that you see there, or is the only thing that you see in a high degree of endemic? Yes. Others, if you want to add in, you can write in the chat box or speak up. High degree of threat. Yes, those are the major aspects. You know, so this picture is not so clear because it's from a really old paper, uh, which is from 2000, like around the time most of us are born. So what they had done was they have had maps of different things. You know, like one map had number of species, like Mrinal has pointed in the chat, and then the other map is of the degree of endemism that is present in each of these areas, and the relative amount of threats, mostly from humans. And there are non-human threats also, but mostly from humans. So they try to overlap all of these together and then find out the regions which where all of these three are overlapping, and they call those areas as biodiversity hotspots. Like these are the hotspots of you know species and all of these areas, all of these aspects like endemism, to threat to their existence. So how many biodiversity hotspots are there in India? Can someone tell me how many biodiversity hotspots? Four. Okay, can you just go through what all are there? Which four are there? Indo-Burma, and this is a really old paper. Is a really old picture. Some of the hotspot has been have been modified. They have been split up, and few more have been added from the time. Okay, someone pointed out Western Ghats. Let's go. Let's start from the south. Okay, Western Ghats in Sri Lanka. So this is one. Okay, let me just write it down there. Okay, Western Ghats. Yes, Western Ghats is one. Okay, Indo-Burma is two. Let me just change the color. So Indo-Burma is two. Okay, Indian subcontinent ends here. Okay, so there is no other hotspots in this map that fall in India. So currently, according to this map, we have two hotspots, but now it has been upgraded into three, one comprising the Himalayas and the other comprising the northeast states that comes here, and then third being the Western Ghats. Okay, so especially because these areas in India also have a lot of species in terms they are endemics. If you have heard of frogs or other groups also, birds also, there's species that are present nowhere else in the world, and they're just present small areas in Western Ghats and not anywhere else, and they have a high degree of threats. And then these areas also have a lot of species like okay. Can you see something similar here to the previous plot, sorry, the previous map? Just have a look at this map. Where are all the areas that are shaded in red? So for this thing, we had three things, right? One is higher species richness, higher degree of endemism, and higher degree of threat. Okay, just have a look at that this plot now. So the ones that are shaded in red or yellow, sorry, uh, are the regions that have higher species richness. Here, vertebrates. Okay, uh, some of these areas coincide with biodiversity hotspots. So, if you see, what pattern are you seeing here? Can someone say what is this plot representing? So, it's talking about species richness, the number of species of vertebrates or the ones that have vertebra on a world map. So, the darker the area or the red, so it says the more number of species, the lighter or the bluish colored, the very few species present in that area. So, what is one interpretation that you can take from this image? Tropical areas mostly, the equator has higher species richness. The equator, right? So if that's the equator, you see most of the species, higher species richness, you know, close to the equator. So you might have Tropic of Capricorn somewhere here, Tropic of Cancer somewhere here. It's not accurate, but somewhere around there. If you see, tropics and subtropical regions, have higher species. Okay, compared to temperate regions. Why do you think so? So this thing that you are seeing, you are seeing a gradient in your number of species that you are seeing. So this gradient is called latitudinal gradient. So this is 0 degrees latitude. If you know equator is 0 degrees, we have 23 degrees. I think Tropic of Cancer, similar to 23 degrees here. You there the lower latitudes have higher diversity, higher latitudes have lower diversity. So that's why this pattern is called latitudinal diversity or latitudinal biodiversity gradient. Okay, this is seen in across all the taxa, be it animals, be it plants, be it insects, fishes, frogs. Most of the taxa have this gradient. Okay, so people just didn't stop like trying to say like, oh, it's there. So what? So they were trying. People were curious. Why? Why do we see higher species in the tropics not in the higher latitudes or temperate regions. So they ended up proposing, different set of hypotheses. One of those hypotheses is, geographical area hypothesis. Okay. Uh, so this hypothesis suggests that, tropics have higher area so that, it has higher species richness compared to, temperate regions, which has relatively lower areas. Yes, you're right. Larger area. So the area might not be just the current area also, but historically, when the land masses were kept put together, because much of the species have been evolved over time, when the land mass were together, much of those land masses were hypothesized to be, according to this hypothesis, much of those land masses were in the equator region. So that area had tend to have higher species. And the other hypothesis that was proposed was evolutionary speed hypothesis. It says that where there is higher temperature, it leads to uh, higher evolutionary speed, due to diversification of the due to higher speciation. So if you see where is the area, where are the areas with higher temperatures? So those are mostly the tropical areas. So in the tropical areas, as there are higher temperatures, that leads to much more higher divergence, and then have those species tend to have higher evolutionary speed, and thus you have species speciation happening much more faster in the tropical areas than the temperate regions. So you have higher species that are accumulated in the tropics. That's why you see higher species in the tropics, what you see currently, compared to temperate regions. And there is another hypothesis that talks about, you know, uh, how species are present in an area. In this example, let's say there are three different areas. Let's say three different forests. Forest one has fire once in 30 years. Forest two has fire once in 3 to 4 years. Forest three has fire every year happening. So if I want to go and understand what will be the number of species present in that area, so which site I might conclude has say has higher species. So did you get the question here? So if I go and sample three different areas with different fire regimes, the one with lower fire regime, one with that happening once in 3 to 4 years, one that in every year. Which of these sites, yellow, blue, and red, I will find the highest number of species of plants. Let's say I'm looking at plants, for example, or you can think of any example: birds, insects, mammals, anything. So someone said yellow, uh, intermediate disturbance hypothesis. Okay. Any other answers? Which one do you think has higher species richness? Yellow. Oh, can one of you defend your answers? Give why do you think it's yellow? Someone said it's intermediate disturbance hypothesis. Yellow or blue? It's not multiple choice. It's more like a single answer. So uh, you're trying to play it safe. I get it. So the thing is, it depends. If I say this is it's someone said it's intermediate disturbance hypothesis, then I will say let's say So the fire happens, once in 30 years. So that is, you can call it's low frequency. The fire happens, not very frequently. Compared to here, fire happens very every year. So which is, much more which very frequently, not so frequently. Once in 3 to 4 years. It's like intermediately frequently. It's like so. So this is kind of, blue is intermediate to high frequent fires versus not very rare fires. So yes. So the thing that you will see here is forest that has, fires intermediately, or here, let's say in this example, once in every 3 to 4 years, you tend to have, higher species richness of whatever taxa plants, mostly in this example. It's blue because yellow may be a moist deciduous forest. That can be a reason also, because if it rains much more, the chance of fire also might reduce, or yeah, humans might be also contribute to, reduction. So yeah, that can be one of the reasons why there might be few fires in that area. So uh, so the basically what does it suggest is, what does this intermediate disturbance hypothesis say is like, the sites that are frequently disturbed versus that are not frequently disturbed may not have much species than the ones that are, intermediately disturbed. So the intermediate disturbed species has the sites has the higher number of species, because there might be different things that might come into play here. Like at low disturbances, competition might be driving, a lot of species to extinction, and then only few species that can compete in that area tend to stay up, while in highly disturbed sites, only few species can stay in that area. Okay, if you go back to the same example, let's say in a site, fire once in 30 years. So you can say that if the area has the forest has reached the climax stage in the successional history, or the species that are present are dominant, there's one species that is dominant. It competes with all other species that are growing, and then you have very few species that are sustaining there. But if the fire happens every year, so whatever the plants that are growing from their, seeds, they burn up every year, and then you might not have, a lot of species. And then when we have intermediate amount frequency fires that are in intermediate frequency, what happens is, some of the plants can grow taller, and some of the plants can grow once the fires burns up. So there's this thing that happens. So both of the species, those that are fire tolerant, those that take advantage of fire and come up, tends to grow together, and then there is no much competition between them, because if the once the time for the competition starts up, there is a fire, and then it just removes the competing species. So at the intermediate levels, you have higher number of species. Okay, so now we till now we went through what is biodiversity, how do we measure biodiversity, and then what are the different processes, that we see maintain biodiversity here, and why should we conserve biodiversity. So can you please answer, this is mostly most likely the final slide. So yeah, can someone please tell, why should we conserve biodiversity? It's there. Why not leave it as such?
So every species is connected to every other species by the flow of matter and energy.
Yes,
And network of life form protects life forms. That's why we have to conserve biodiversity. The more the biodiversity, the healthier will be the ecosystem, and all the
yes,
species will remain protected.
yes. Anyone wants to add anything? what Prashanta has said. So there are multiple reasons that why we should conserve biodiversity. To prevent species extinction, yes. What happens because of species extinction is what Prashanta was trying to say, because because these species, every species is connected to each other in the food web or ecological structure, so they each have their own role. So if one of the species main doing one of the crucial roles goes extinct, that whole structure that we that it is a part of might not be able to sustain itself. So there are multiple reasons whether which you people have pointed is or of more of ecological reasons. There can be scientific reasons why should we conserve biodiversity, because people wants to learn or to get inspired from how different things go on. Let's say, for example, if you want to build taller buildings, you might be inspired from how termite builds their termite mounds with effective ventilation. And there are cultural, there is scientific reasons for biodiversity. You have economic reasons because you get a lot of economic benefits. Let's say, for example, uh, bees pollinate much of the crops that we get. So once most yeah, by protecting others, we remain protected. So much of the crops, most of the crops are pollinated by some sort of insects. So it has some economical benefits. It has cultural benefits. Let's say, for example, elephants, tigers in India. Most of the species in India have, cultural benefits. Even if you go to northeastern parts of India, where hornbills are, hornbill casks are an integral part of their, the local culture. Similarly, reasons why we should conserve benefit biodiversity is what we are discussing, and recreational. If you go to a tiger reserve, or you just want to go hiking, so you watch biodiversity or you just go around. So there are different reasons why we should conserve biodiversity, especially in this era, because we are causing the we are when we are the major reason for the loss of biodiversity. Okay, and yeah, so yes, that's it for today. Okay, so if you guys have any questions, do let me know so that I can, in whatever 10 minutes, 15 minutes, I can go over again. So if you want to, have a look at the calculation again, it will be there on this, on this channel on YouTube, so that you can go back. I will upload it by tonight or tomorrow, so that if you need to look at the calculations part again, you can just go there and then check it out. So yeah, and I just like to point this thing out, as Prashanta was saying earlier. Uh, Not always high biodiversity means, you know, high ecosystem. We just wrote earlier. Just a minute. High biodiversity doesn't mean always it's a healthy ecosystem, but most of the time it implies it's a healthy ecosystem, but not always. Like, say, for example, if you're an invasive species, so all the invasive species come together, then that system might not be healthy, because there you're seeing a lot of diversity there, but that's that's not might not be what is the true ecosystem that you have to see, because all of the species that you are present there are invasive or non-native to that area. So, but in most of the case, higher diversity means tends to stand true for that statement, but it's not the universal case. Just keep that in mind. Yeah. So that's all from my side. If you have anything, just let me know. Any doubts or any other from the calculations or anything? No? Yes? I hope I, I think you could understand most of the parts. Then, yeah, in that case, I think we can leave. Yeah, thank you for joining this class. Hope you get to learn something interesting and new. Thank you. Yeah, you're welcome. And if you want to go back to the class, just refer to the